Hello and welcome to this video podcast for evolution and diversity. Up to this point, we spent a lot of time talking about evolution, speciation, phylogenies, and how organisms were, in general, connected with each other. So for most of the rest of the semester, we're going to start looking specifically at types of organisms. And over the next several days, we're going to focus on bacteria and archaea. And in this podcast, we're going to talk about the adaptations that bacteria and archaea have derived to allow them to become the organisms they are today. We're going to begin with an evolutionary relationship between the three domains, bacteria, archaea, and eukarya. And then we're going to spend quite a bit of time talking about the structure and organization of bacteria and archaea. And then we're going to close with talking about some of the main differences between archaea and bacteria. And some of those differences we'll have already highlighted in part 2 here. So let's go ahead and get started. So we've drawn this phylogenetic tree before, and so let's just draw it again here. And we know up here we have the eukarya, and we'll spend a lot more time talking about those in the next unit of material. But we're going to focus today on the domains of bacteria and archaea. So as we look at this, let's review some of the things we learned about phylogenetic trees. So the first question I would like to ask is, are archaea more closely related to bacteria or eukarya? So I'll give you a moment to think about that, and if you want, you can hit pause while you're considering the answer here. So the answer is that archaea are more closely related to eukarya. We'll learn in a moment that bacteria and archaea look very similar. They're both single-celled organisms. But from an evolutionary perspective, archaea and eukarya, they share a common ancestor together here before they share a common ancestor with bacteria. Because of that, archaea and eukarya are more closely related. Also remember that these nodes are rotatable. So we could have just as easily wrote eukarya here and archaea here, and it would have meant the same thing in regards to the phylogenetic tree. Now, before I ask the second question, I want to point something out for you, and that is archaea and bacteria are often called prokaryotes. In general, we don't use the word prokaryote because it implies a false relationship that archaea and bacteria are more closely related than eukarya. And that's not the case. But when you cluster them together by unifying them under this one term, it does give that impression. So in this course, and the rest of your biology courses, we will more often use the terms individually as bacteria or archaea, or if we're talking about both of them together, we'll just say bacteria and archaea. Now, the one time that it's appropriate to talk about them as one group is if you want to talk about unicellular organisms or organisms without a nucleus. Pro means before, karyote means karyon, and karyon is a word that means nucleus. So really this means no nucleus. So if you want to talk about all organisms that don't have a nucleus, then it would be appropriate to call them prokaryotes, because that's what bacteria and archaea are together. These are also organisms that in general lack organelles. So if you want to talk about organisms that lack organelles and you wanted to cluster them into bacteria and archaea, that would be okay. I would still prefer you to use the word bacteria and archaea as opposed to the word prokaryote. Okay, so with that in mind, my next question is, do bacteria and archaea form a monophyletic group? You can think about that, and if you want, you can hit pause while you think about it, and then hit play again to see what the answer is. So the answer to this question is no. They do not form a monophyletic group. Remember, monophyletic group includes the two taxa plus their common ancestor. Bacteria and archaea do have a common ancestor, which is here, but this common ancestor is also the common ancestor for eukarya. And therefore, you cannot say bacteria and archaea are a monophyletic group. You could say bacteria, archaea, and eukarya themselves form a monophyletic group because it would include the three taxa at the top plus the common ancestor of all three of them. So let's move on to the second objective for today, and that is describing the structure and organization of bacterial and archaeal cells. And so what I'm going to do here is I'm going to draw a general bacteria or archaeal cell, and I'm going to identify parts of these cells. And for the most part, whatever I indicate here, you can assume could be found in a bacterial or an archaeal cell, except for when I point out some differences. And there will be some differences that I will point out. So if you remember that primordial cell that we talked about very early in the semester, where we talked about how this self-replicating RNA molecule was encircled by a lipid membrane. And so what we're thinking about here with a bacterial or an archaeal cell is going beyond that primordial cell and adding a few adaptations to it to make it a much more functional cell. In fact, an incredibly functional cell. You'll learn throughout the next several lectures that the diversity of bacteria and archaea is amazing. And the many different things that these single-celled organisms can do is equally amazing. So what I drew here first is just a plasma membrane. And these are just the phospholipids that we talked about earlier in the semester with the primitive cell. And they will hold within inside of this plasma membrane the cytoplasm. The cytoplasm is the thick solution of salt, water, and proteins inside a cell that is surrounded by the plasma membrane. Next, let's talk about the chromosome within the bacterial cells here. We've talked about the chromosome before, but let me remind you what it is. It's a structure in a cell consisting of a long molecule of DNA. So in humans, we have 46 chromosomes. And remember, whenever we've drawn them before, we think of them as looking as a single linear piece of DNA. And again, in humans, there are 46 of these. Bacteria and archaea are quite different. They only have a single one chromosome. And this single chromosome is circular. There's always some exceptions, but most bacteria and archaea have circular DNA. And again, remember, in bacteria, there's just one of these. Before we move on, I want to point out one rather spectacular thing about bacterial's chromosomes. It's also pretty spectacular in eukaryotes, but we're going to focus on bacteria and archaea here. This is a picture of a bacterial cell. And this bacterial cell has been lysed so that all the chromosomes have spilled out of the bacterial cell. And so you can see within this tiny bacterial cell, all this DNA is packaged into it before it was lysed, right? So any bacterial or archaeal cell that you find will have approximately this much DNA in it in relation to its size. So the impressive thing is all of this DNA is packaged into this cell, and I should also point out, and we'll talk about this term in a moment, but it's all packaged within this cell in a much smaller structure. Not a nucleus, and we'll tell you what that structure is in a moment. But all of it is packaged into that small little area. And it's not just like when, I might be packing for a vacation, I just cram everything into my suitcase, and when I get to where I'm going, it's all wrinkled and unusable. That's not what cells will do. They package all this DNA into this tiny cell, into this tiny area within the bacterial or archaeal cell, and it's usable, right? It's not just shoved in there hoping for the best. It's put in there in a very precise way so that the cell can use that DNA to make proteins at the right time. So what is this tiny little structure here? That tiny little structure is called a nucleoid. And so the nucleoid it's not a membrane structure, it's just an area within the cell. And this area within the cell is where the chromosome is. So it's reminiscent of a nucleus, but it's not, because a nucleus has a membrane around it. But it is clustered in one part of the cell, and so that's what we call the nucleoid. Another thing that is important to point out are ribosomes. And ribosomes are going to be found throughout different parts of the bacterial or archaeal cell. But let's go ahead and just identify 3 of them here. A cell would always have many more than just 3. These are structures in the cytoplasm that contain RNA and protein. And the job of the ribosome is to make proteins. That's what it does. And how does it make proteins? Well, that would be many lectures to explain exactly how it makes proteins, but I'm going to just tell you real quick in general how it does it. This is the ribosome. The ribosomes always contain a small subunit and a large subunit. Each of these subunits contain many RNA molecules and many proteins. But it will take a messenger RNA and send it through this ribosome to produce a protein of some specific nature. It's important to point out here that in bacteria and archaea, the size of the ribosome is approximately 70 Svedberg units. Don't worry too much about what a Svedberg unit is. It's a unit of density that is just how we measure large structures like ribosomes. So that's about how big it is in bacteria and archaeal cells. So I'm going to put B and A here for bacteria and archaea. However, in eukaryotes, it's a little bit bigger. It's 80 Svedberg units. And I only point that out right now because this is a way that we can design antibiotics to attack certain bacteria without those antibiotics attacking our eukaryotic cells. Okay, I have other things I want to label in this, but I don't have room. So I'm going to erase everything I have here so far, and then we'll add some more to this picture. While I was talking about the chromosomes, I should have pointed out that there are other pieces of DNA in the bacteria and archaeal cell. And these pieces of DNA we call plasmids. These plasmids are smaller circular pieces of DNA. So compared to the chromosome, it's much smaller, and because of that, it will contain fewer genes in it. But the genes it does provide lots of different functions. So every plasmid will have a different function, and a particular bacterial or archaeal cell will contain several of these plasmids, each with a different set of genes on it and a different function. Real quick, some of the functions include antibiotic resistance. Some of these plasmids produce toxins. So these first two are related to the bacterial or the archaeal cell's defense. Some play a role in replicating, and I'm just going to put REP here for replicating the chromosome. Some are involved in fertility. Some digest certain rare substrates. And the last thing I should put here for a function is outside of the bacterial cell, we use plasmids all the time in the lab to clone genes. And I would suspect many of us have benefited from the ability to clone these genes. For instance, so if you know anyone who is a type 1 diabetic who takes insulin, so that insulin is made, real briefly, by taking the plasmid here, putting the human gene of insulin in that plasmid, and then placing this inside of the bacterial cell, and that bacterial cell will now make insulin protein. It'll make human insulin protein. And then we can purify that human insulin protein because we can make a lot of it in a big vat of bacteria, and then that is packaged up and sold to people who need insulin to treat their diabetes. One other thing I should say about plasmids is just to introduce another term here related to fertility. Bacteria and archaea have the ability to conjugate with each other so that they can pass genes from one cell to the other cell. And we'll talk more about this in the next lecture, or maybe it's the lecture after that. But the only way they can do that is that they have to connect. And they connect by forming what is known as a sex pilus. And then this sex pilus serves as a transfer mechanism of taking DNA from one cell and sending it to the other, or parts of that DNA from one cell to the other cell. All right, two more things I want to talk about here are the cell wall, and I'm just going to identify it now, and in just a little bit I'm going to go over it in much greater detail. So this is a cell wall. So this is in addition to the plasma membrane, and it will serve a role in protecting the cell, which we'll discuss in a moment. The next thing is this structure around the bacterial cell that surrounds the cell wall, and this is called a capsule. A capsule is a gelatinous layer allowing the cell to adhere to surfaces. So essentially, it allows it to stick to things. And this protects the cell from the immune system, and it also protects the cell from being digested by phagocytic cells. This is a very important adaptation for bacteria to evade destruction by the immune system and allowing it to survive. Okay, I guess there are a couple other structures I want to point out. And so bacteria and archaea can also have structures sticking out on the outside of it. And they look like spikes. And I guess in many ways they serve as a spike because they protect the cell. And these structures here that I said look like a spike, they're called fimbriae. Like the capsule in green that I showed before, fimbriae also help in adhering to the two surfaces. So another very important adaptation. Another structure I want to point out that some bacteria and archaea have, not all, is that some will have a flagellum. Now this flagellum is really important in cell movement. It helps the bacteria move. But it's an extraordinarily amazing adaptation for bacteria and archaea cells. And though it can't really be seen here too well, within the structure of the flagellum is an elaborate protein motor. And this protein motor functions very much like a motor would, and it causes this flagellum to rotate and spin around in order to move. So I wanted to show this quick video of what the motor looks like for the flagellum. You don't need to memorize anything that you see in this video. I just find it to be a very good representation of what's happening with the flagellum. So what you can see here is we're looking down on it. This is from the outside of the cell, and you can see the flagellum moving out here from the cell. And on the underside here is the actual motor, and we'll see that in just a second. Now you see many parts here that are made of proteins, but they function just like a regular motor would, turning this around in a particular motion and spinning an axis of some sort here in the middle here to flip this tail around and around and around. So I'm going to let it play for just a little bit here. And I'm going to stop it here. And as you can see, this tail was spinning, and it will move in a certain direction, as I said before. And when they record this, it can spin at about 100,000 RPMs in that one direction, which will then propel the cell in that direction. And then if it needs to move and go in the opposite direction, it can immediately reverse it and begin spinning 100,000 RPMs in the other direction. So this molecular machinery that is the motor for the flagellum is truly an amazing structure that the bacterial cells adapted over time, and it made these bacterial cells with this ability to move rapidly in a particular direction more fit and more likely to pass on this trait. And as we've talked about with everything in evolution, we're certainly not saying that this very elaborate motor wasn't there in one generation and in the next generation, voila, this whole motor was there. This would have been a stepwise process of building this motor over many thousands of years. Okay, let's go back to the whiteboard. A very advanced adaptation that some bacteria and archaeal cells have. All right, just a couple more things I want to point out. And it seems like I said a couple more things for the past maybe 5 or 6 things, but I think we're down to just a couple more things I want to point out. And one of them is a cytoskeleton. When I was in college, I was taught that bacteria do not have a cytoskeleton. This is something only found in eukaryotes. And while for the most part there's a lot of truth to that statement still, we have discovered proteins within bacteria and archaea cells that behave like a cytoskeletal protein. So we're going to call these primitive cytoskeletal proteins. And there's only one that I want you to know, and that is this protein called PhytZ. And it behaves very much like actin does in eukaryotic cells. And what PhytZ will do is it will help the cell divide. So if we have our cell here, and we have our DNA, our chromosome here, that chromosome is going to divide. And when it divides, you're going to have a bacterial cell, a little longer, with chromosome on either side, the copied chromosome. But the cell needs to split. And so what PhytZ does is it will form a ring around the inside of this cell and pinch the cell together and squeeze it apart in such a way that it results in 2 newly formed cells. This process of dividing a cell in bacteria is called binary fission. In eukaryotes, you'll learn that the process of dividing a cell is either mitosis or meiosis, depending upon which type of cell you're looking at. But in bacteria, they go through a process called binary fission, and they do not undergo mitosis. And you'll learn much more about that later on, and in particular in cell biology and genetics. Similar to the story of cytoskeleton, when I was in college, I was told that there were no organelles inside of bacteria. And still, that is a true statement in the sense of what a true organelle is. However, over the past several years, these primitive organelles have been found in some bacterial species, and Archaea species as well. And these include some primitive organelles that store calcium ions. They help organize enzymes for making carbon molecules. And this last one, which I'm almost out of room here, but I want to write it down because it's kind of cool. And these are called magnetosomes. And they hold magnetite crystals. And so they form these very high concentrations of iron-rich magnetic particles. Okay, I promise this is the last new structure I want to tell you on this picture here. And after I tell you that one, then we're going to go back and we're going to talk about plasma membranes and cell walls in more detail. But this last structure here include photosynthetic membranes. And these are only found in certain bacteria, and we call those cyanobacteria. And as the name implies, these bacteria can undergo photosynthesis. And this is what one of those cyanobacteria look like, a bacterial cell that has many of the same features we just talked about, but they also contain elaborate membrane structures that allow for photosynthesis. As I mentioned, I wanted to come back to the plasma membrane as well as the cell wall, but let's talk about the plasma membrane. And I really want to talk about what makes the plasma membrane different in bacteria compared to Archaea. So the plasma membrane, for the most part, looks the same in bacteria and Archaea as far as a general structure goes. So remember, they would look something like this, with the polar phospholipid head here, with the phosphate group at the top. That is polar, and as we said, that makes it hydrophilic. And then these fatty acid tails here, which are nonpolar, and we would call those hydrophobic, just as a bit of a review. What's different between bacteria and Archaea is in the way that they link up here with the fatty acid tails. So I want to show you now just a couple images of what a bacterial phospholipid would look like compared to a Archaea phospholipid. So what we are showing here is on top the bacterial phospholipid, and also it's what we would see in eukaryotes as well. And on the bottom is the archaeal phospholipid. Both of them have this phosphate group, and they both have a glycerol group. One's a D, one's an L. That does make them different, but don't worry too much about what the difference is between a D and an L form. But what I would like you to know is that eukaryotes and bacteria have an ester linkage here with the fatty acid tails, whereas the Archaea, they have an ether linkage with the fatty acid tails. Don't worry too much about what the difference is between an ester and an ether. You'll learn more about that as you progress in chemistry, and we'll touch on it a little bit in cell biology. But what I want you to know here is that eukaryotes and bacteria have an ester linkage, and Archaea have an ether linkage. That's what you should know. And so the next question I want to address is, why is this important? Why is it important that Archaea have an ether linkage? And we'll talk about this later when we start talking about some of the other distinguishing features between Archaea and bacteria. But for now, what I'd like you to know is that Archaea live in extreme environments. This ether linkage allows that plasma membrane to be stronger in these extreme environments, like elevated temperatures, for instance. Now let's think about our phylogenetic trees here. So let's think about a phylogenetic tree, thinking about these differences in the fatty acid linkages. So we have our bacteria up here, and our Archaea here, and then our eukaryotes here. So in this common ancestor of bacteria, Archaea, and eukaryotes, there would have been phospholipids with an ester linkage. This would have been an ancestral trait. We still see this ester linkage in bacteria and eukaryotes. During the evolution of Archaea, they developed the adaptation of an ether linkage, which allowed them to survive in those extreme environments. All right, now I'd like to talk about the cell wall. And I want to tell you how it is different in different kinds of bacteria first, and then I want to tell you how it's different in Archaea. Bacteria are often categorized based upon whether or not they are gram-positive or gram-negative. I'll show you a picture in just a moment, but the main difference between a gram-positive and a gram-negative bacterium is that the gram-positive has more of this carbohydrate called peptidoglycan. And when you stain a cell that is a gram-positive cell, it turns a brighter purple. And that's because the stain is specific for peptidoglycan. Gram-negative bacteria, they still have peptidoglycan in them, but they have less peptidoglycan. And when these cells are gram-stained, you get a lighter purple. It looks more like a pink, I guess. And real quick, this is how the gram stain would work. You would put the bacterial cells on a slide like so. You would stain them with a dye called crystal violet. You would then add iodine and then wash all of that away with a decolorization chemical. And what that leaves behind are the gram-positive cells, stained purple, and then the gram-negative cells that are not stained. And then they are counterstained with safranin that helps bring out the color in the gram-negative cells so that when you're looking at a slide that has a mixture of gram-positive and gram-negative cells, you can distinguish between them fairly simply. When identifying a bacteria, this is one of the first steps that is carried out. That way you know if it's a gram-positive or a gram-negative cell. So let's come back here and think about the structure of the cell wall, and that'll help us understand why it's stained so much better. So this is a picture of what a gram-positive cell would look like, and you can see this very thick layer of this peptidoglycan on the outside of the cell. So you have the plasma membrane, and then below the plasma membrane, this is the inside of the bacterium. Okay, so on top of the plasma membrane is this very thick cell wall made up of this peptidoglycan structure, which is a combination of carbohydrates and proteins. So this is what a gram-negative cell wall would look like. You still have the plasma membrane, and then on top of it is the cell wall. And the cell wall consists of the peptidoglycan that we saw in gram-positive cells, except it's much thinner. It also has an extra layer of a membrane on top of it. Because of that, gram-negative cells typically have a much higher lipid component to them. And they stain less well because there is less peptidoglycan in these cells. Another thing I would like to point out about gram-positive cells, as shown here, is that they are less resistant to antibiotics. In fact, a lot of antibiotics are designed to disrupt the production of the peptidoglycan cell wall. These are often our first line of antibiotics. However, these same antibiotics are not very effective against gram-negative cells. So as a result, gram-negative cells tend to be more resistant to antibiotics. So people who have a gram-positive infection are typically easily treated with antibiotics. Of course, not always. But somebody with a gram-negative, they're less likely to be treated effectively with an antibiotic. So to summarize, bacteria, they have cell walls, and those cell walls are either gram-positive or gram-negative. And it's based almost entirely upon how much peptidoglycan is found in that cell wall. So let's now talk about the archaeal cell wall. The biggest difference between archaea and bacteria in their cell wall is archaea have no peptidoglycan. But they do have something called a surface layer protein. This is a fairly rigid array of proteins on the outside of the cell. And this provides chemical and mechanical protection. This is likely an adaptation that has helped archaea survive in those extreme environments. I should also mention that this surface layer protein, which is often just called an S layer, is found in some bacteria. Now the bacteria that have the S layer level of proteins, they also still have the peptidoglycan in them as well. It's just an extra layer for some bacteria. Now I would like to talk a little bit about bacterial cell shape. And when we categorize bacteria, we often place them in one of three shapes. The first is circular. The second is rod-shaped. And the last is curved shape. And there are variations within these, but these are the three broad categories. Circular are what you might expect. They're very circular, very spherical. Some Examples of this include different species of the genre Streptococcus. One in particular, Streptococcus pyogenes, is associated with things like strep throat, scarlet fever, necrotizing fasciitis, the flesh-eating bacteria. Another example would be Staphylococcus aureus. This can also lead to various human pathogens, such as something as minor as acne and all the way to things like pneumonia and toxic shock syndrome. Now, I'm sure this is going to be shocking to you to find out that bacteria that are classified as rod-shaped have a shape that looks like a rod. Some examples of bacteria that are rod-shaped include Bacillus anthracis, and this is the species that would produce the toxin anthrax. Other species that you've probably heard of are things like E. coli. This, of course, includes a variety of E. coli, including the ones that live in our bodies right now and causes no harms, all the way to various strains of E. coli that can make us very sick. Another example of a rod-shaped bacterium is a mycobacterium. And this would be associated with things like tuberculosis or leprosy. Lastly, I want to talk about the curved-shaped ones. Now, these can have various shapes, and some of them will look like the rod-shaped ones, but they might be a little curved, or they might form a spiral. An example of the ones that look more like a curved rod here would be various species from the genre of Vibrio. These are often associated with foodborne illnesses like undercooked seafood. The ones that are spiral-shaped here include bacteria from the genre Spirochaeta. And these species from spirochete genres are linked to diseases such as syphilis and Lyme disease. So let's go ahead and talk about the differences between bacteria and Archaea. We've already talked about several of these, and we'll bring up more as the next few days go on. But let's just make sure we keep a running total here. So the first one is that bacteria have a phospholipid with ester linkages, and Archaea had a phospholipid with ether linkages. We also talked about the differences in the cell wall. Bacteria have cell walls that have peptidoglycans in them. Now, the gram-positive bacteria have many more peptidoglycans compared to the gram-negative ones, but they still both have peptidoglycans as a part of their cell wall. And Archaea lack peptidoglycan, and their cell wall is based exclusively on having this surface protein layer that we call the S layer. And this third one is a difference in ribosomes. I did talk about how ribosomes were different in bacteria and Archaea compared to eukaryotes, but I didn't really focus too much on the difference between bacteria and Archaea, but let me do so now. And all you should need to know is that they have different proteins and RNA molecules. And the last one I want to say is something we mentioned when we were talking about the cell wall and the phospholipid plasma membranes. And that is that Archaea live in extreme environments, and bacteria don't live in extreme environments. And we'll come back to this last point later on. There are also differences in the way that these two domains replicate their DNA or express their genes that we didn't talk about, and we probably won't talk about in this class, but in other classes as you move forward, you will discuss DNA replication and gene expression. All right, that's all I have for today in this video podcast. If you have any questions at all, please make sure you let me know. If not, I will see you in class. Bye.